identify this resistant population of aquaculture species. Because this is not details, so I means here I want to review in some papers, several papers, maybe 100 papers, because in this topic is very few examples in aquaculture, maybe different with agriculture or animal husbandry. So first, I will organize my presentation today. First, I will make a background, make a context, what I'm trying to talking about these topics, and then I will explain a little bit about what is microsatellite concepts, how they are isolates microsatellites, and how to apply it, and I will make a conclusion in the big picture of science. And the first, I'll make a, maybe you not too familiar with aquaculture. If a aquaculture company, they need the seeds, good seeds, good water quality management, good feeds, and over several period of time they will help us fishing these aquatic organisms to make money. So we call aquaculture is culturing aquatic organisms to get profits. This is a simple definition of aquaculture. So my focus here is in seeds, how to make a better genetic makeup of our seeds, and of course our food stock, to get better survival rates. I told you before, why this kind is so very important, because viral disease remain unsolved. This is WSSV outbreak. This from East Java. I have this. This is in Aceh, because I work in marine department. So this is my own experience. It's very difficult to treat microcephalic fires. But then this my data too. Bacterial diseases are everywhere. Bacterial infection. I will give you this example. Mycobacterium and Vibrio attack this tilapia. Then this Streptococcus ciniae attack the tilapia, saltwater tilapia. This Streptococcus ciniae. This is a coccus. Yeah. And then a solution in genetic perspectives. We'll give you maybe this. All of you know that conventional hybridization. As Professor Morris said, that conventional hybridization is need long time in conventional hybridization, and transgenics is publicly unacceptable. I don't see this. I just want to make this strong message about this transgenic. Maybe this mock you as a geneticist, and then it's better to compromise between conventional hybridization and transgenic is microsatellite assisted selection. So we choose this kind of approach. Then what is microsatellite concept? You see in everyday life, you see this son, this handsome son, these handsome guys. Who is his father? This Charles Prince say, "It's my lovely son." It's my last baby. It's, Professor Bush say that, and I say, "Don't agree with this. That's okay. It's really for me." Okay. And this for our daily life. And then a code in all living things stay the same. A, T, G, C. All of you know about this. And this I give you this. Maybe I want to try this to make you understand. Maybe some of you don't know about this information. And this what is microsatellite. You see here T several times, AAA several times, and AG several times. I will give you that this AG I mean a repetition of several times of the same sequence of DNA here. AG. This is microsatellites. Yeah, this microsatellites. So this beside this right side and the left side of microsatellites is flanking region. So we make a primer from this flanking region. Primer attached this fourth part of the sequence. Thus from this we can amplify in polymerase chain reaction. I think it's familiar for you. So because I give you a simple conclusion. This microsatellites are repetition of DNA sequence. And maybe if you want to know what the more complicated ones, definition of this macros you can see in some review of journals. And I'll give you these examples. Maybe I'll give you example this. Short DNA make sick, some kind of sick for fish. And this long DNA make a fish healthy. Maybe I just make to make oversimplify. So I'm sorry, I'm too oversimplify. Why it happens? Because microsatellite function is really important to regulate gene activity. I mean, that's microsatellites that place in the intron in the gene. It's important, very important. We say that there is two types of microsatellites. The microsatellite that outside the genes, maybe some of you call this junk DNA, but this junk is this junk is important. This one, and first is regulates the metabolic process and stabilize in chromosome structure. Many of microsatellites place in telomere of chromosome, so it stabilizes chromosome structure. And then how the methods? Maybe this I oversimplified again the methods. So extract microsatellites from the fish, and then find their pattern of microsatellites using DNA sequencer and make a pre-map here. I told you before, this pre-map, the left side and the right side of the microsatellite. We choose first the better one from the primer, and then we multiply them in PCR polymerase chain reaction, and then we make several samples, evaluate in population. Where is the sick and healthy one? And then because these have different size of DNA, this short and this long microsatellites, and then with that after we amplify and put in gel electrophoresis, we can see here this different. This is for black tiger shrimp. This one. Yeah, you know here. This is for health sick one. This is for healthy one. It's already known from 2009 papers. It's very rare for these topics. And application, I give you two examples here. If you want to know, I have an appendix of this some kinds of research. And such white spot syndrome virus resistant shrimp by Mukherjee and Mandal 2009. Even this is a pioneer of this research. This is quite successful. We see here this is white spot syndrome virus. Yeah, it is in Aceh, in ponds in Aceh, and also not only black tiger but Chinese shrimp. Now this in fact Chinese shrimp in China. This is by Dong et al. 2008-2009. And then I give you this here molecular breeding. This choose from highly resistant female, the crossbreeding with wild male, and gave 163 juveniles fit with virus. I mean the shrimp that have a positive virus, and then spread the sick and the healthy one, and test with 50 microsatellites, and after it we can figure out in the gel electrophoresis. This one you see here, this double band, this single band. This double band is healthy. This single band is sick shrimp.
5 more minutes.
Oh, definitely okay. Okay, this second examples in Japanese flounder. This is quite successful. Many people very appreciate this work because they worked this for 3 years. This one is sick one, then this healthy one. This flounder attacked by leprocystis virus. It's become very ugly. This is become very ugly, and they breed the sick family and healthy family and check for the children and crossbreed with the healthy family and get sick and healthy one, and finally they found that a microsatellite called poly9-8TUF can identify disease resistant population. You see here, it's very different. This healthy one and 61. And counted 147 base pair more responsible for deposit disease resistance. So I conclude here based on only two examples. If you want to know, you just read my papers in the CD. The proceeding conclusion, my conclusion here: high mortality could be prevented by disease-resistant food stocks, and evidence show that microcystrid can detect susceptible and resistant strain to specific disease. Conclusively, microcystrid is powerful marker in selective breeding. So if you maybe you disturb something in my presentation, you just remember this one, this hint, this bottom line. The survivors have the science in their DNA. Thank you very much for your attention.